You are lying in a dark room. Your body is entirely paralyzed, unable to move a single muscle from the neck down. Yet, under your closed lids, your eyes are darting frantically back and forth, and your breathing has become completely erratic. To anyone watching, you look perfectly still, but inside your skull, your brain is currently burning just as much oxygen and glucose as it does when you are wide awake. The traditional assumption about sleep, often called the recuperation theory, is that the body and brain simply power down. We treat it like turning off a machine to let the hardware cool and wipe the operating system clean for the next day. But decades of clinical measurements show that this passive model of rest is completely backwards. Falling asleep initiates the transition into a biologically expensive and highly active alternative state of consciousness. This realization began when early researchers started tracking the electrical output of human brains through the night using polysomnography. This graph shows a standard 90-minute human sleep cycle. As a person transitions from wakefulness, the cycle progresses downward into the stages of non-rapid eye movement, or NREM sleep. At the bottom of this descent, neural synchrony slows. The brain's electrical patterns stretch out into low-frequency, high-amplitude delta waves. Your physical body relaxes, but your muscles still maintain their basic tone. But somewhere between 70 and 100 minutes after you close your eyes, the pattern reverses. The electrical waves suddenly spike, returning to the high-frequency, low-amplitude patterns of a brain that is completely awake. This is the onset of rapid eye movement, or REM sleep. During this phase, a mechanism known as REM atonia engages. Your brainstem sends signals that completely disable the motor neurons in your spinal cord, leaving you paralyzed from the neck down, even as your brain fires at maximum capacity. Evolution programmed our biology to spend roughly a quarter of the night working furiously to generate vivid hallucinations, while simultaneously locking down the physical body so we can't act them out. The trigger for this state is a massive flood of the neurotransmitter acetylcholine. Originating in the basal forebrain and brainstem, it washes upward into the cortex, driving severe neurological arousal. But this chemical flood specifically targets the limbic system near the center. These primitive regions process raw emotion, fear, and memory. Their hyperactivity causes REM scenarios to manifest as intense anxiety or dread. At the exact same time, electrical connectivity to the frontal lobe, the region responsible for logic, drops off drastically. You are biologically trapped. Your brain is generating chaotic emotional scenarios, but lacks the logical capacity to recognize that they are entirely fabricated. If these experiences are so biologically intense, we have to ask why we almost instantly forget them the moment we wake up. The answer lies in a different neurotransmitter, norepinephrine. The brain requires this specific chemical to convert short-term experiences into permanent memories. But during REM sleep, your levels of norepinephrine drop to near zero. Your brain chemically prevents the active hallucination from being recorded to your memory. Dreaming also occurs outside of the REM phase. Light sleepers often experience mundane, thought-like dreams during NREM stage 2, but the primary REM state remains a biological paradox. The brain expends massive amounts of energy to run highly detailed emotional simulations, only to mandate their immediate chemical deletion. Scientific attempts to explain why this mechanism exists are divided. One framework is the activation synthesis hypothesis, which suggests dreams are merely a byproduct of routine electrical maintenance. The brainstem periodically fires electrical bursts, known as PGO waves, into the visual cortex. According to this theory, the sleeping brain is just frantically trying to weave these random neurological flashes into a cohesive story. Another approach focuses on cognitive function. This theory argues that dreams purposefully prime associative networks, allowing the brain to consolidate daytime memories and solve problems without the constraints of waking logic. There is also a strictly evolutionary perspective. Researcher Ioannis Tsoukalis has proposed what is known as the defensive immobilization theory. Tsoukalis points out that the physical conditions of REM sleep share striking neurophysiological similarities with the tonic immobilis reflex, the biological mechanism prey animals use when instantly freezing to play dead and evade predators. Much of early sleep research was built on studying these same mechanisms in animals, tracing the behaviors that dictate survival across different species. Whether it's a random electrical misfire, a memory sorting algorithm, or an ancient predator evasion reflex, the biology of the dream state was deliberately preserved by millions of years of evolution. We can see the necessity of this process by observing what happens when it is denied. A comprehensive meta-analysis from the University of Pennsylvania examined the specific impacts of short-term sleep deprivation across various cognitive functions. This chart shows the researchers' findings. While complex tasks like reasoning accuracy saw only small, non-significant declines, there was a massive drop in simple attention and vigilance. Deprive the brain of its nightly paralyzed hallucinations, and our ability to sustain basic waking attention physically breaks down. We have mapped the chemicals that induce it. We can measure the electrical storms that sustain it, and we know exactly how cognitive performance collapses without it. Yet, the unified biological purpose of dreaming remains an unsolved mystery. Behind the mask of nightly paralysis, the brain begins its most intensive labor of the day.